And then we move to our next speaker, who is Dessislava Veltcheva from Martin Main's lab at the University of Oxford. Yeah, I already see her, so then you can share your slides and she will tell us about fluoroquinolone resistance in Campy.
Yeah, awesome, great. Hi everyone, my name is Dessi. Thank you for the introduction. So I'm from the University of Oxford. I'm in my third year of my PhD. So today I'll be talking to you a little bit about the longitudinal analysis of Campylobacter jejuni's fluoroquinolone resistance among the human disease isolates. So these are still unpublished data. I'm happy to be recorded, but it will hopefully be coming up soon. So just to let everybody know a little bit, I know there's a lot of audience from outside of UK as well. So in terms of the antibiotic usages in the farms, we have had kind of these two main eras, and now we're hopefully going into the third era. So from the 1990s to 2006, we have had no laws in terms of what antibiotics we could use. So I call it the antibiotic era, where we can use antibiotics for growth promoting reasons, for therapeutic reasons. In 2006, we have had the ban on antibiotics for growth promoting reasons. So we come into this kind of era of restricted antibiotic era. So it's still, we can use it for therapeutics, but not for growth promoting reasons. And then in 2022, the EU is now thinking of banning the therapeutic reasons antibiotics as well. But at this stage, UK is still kind of, with Brexit, we still don't know necessarily if we will be following that law or not. And since I am co-founded by the Food Standard Agency, we're really interested to understand what is going on in the antibiotic fluoroquinolone resistance across different strains, and also to understand what could become if we do follow the law or not. So in terms of resistance, Campylobacter jejuni has resistance in different types of antibiotics, especially on fluoroquinolones and tetracycline. But I have decided to focus on fluoroquinolone resistance due to the fact that the correlation between genotype and phenotype is very high. So if you have the gyrase gene mutation from threonine to isoleucine, we have a very phenotypically, we can also see that resistance as well. So from now, whenever I say resistance, I actually mean that I'm using the genotype to predict the phenotype. In terms of antibiotics resistance, we all know Jim O'Neill's report that it is a very big issue. In 2050, it's been predicted that 10 million people will be dying from antibiotic resistance. So this is kind of the reason why I'm interested in this. Okay, so my data is all coming from the PubMLST database, and mainly it is coming from Aberdeen. I'm not sure if you can see my screen, but my pointer, but yeah, I think that's, no, that's fine. So it's coming from Aberdeen, Newcastle, and Oxford. And also we have data all across from the 1997 to 2018. And we have quality checked all these isolates. So all the isolates have, for example, clonal complex data as well. And just to let everybody know, the I is the resistant point mutation, and T, so threonine, is susceptible isolates. So I'll be going over these four main type of analysis, and kind of I will showcase that story behind what is going on. So the first stage is to really understand, to normalize the data. Since, as you saw, some of the years we don't have that much data points, what we want to understand is, if we normalize the data, what is the resistance across time? So the first thing we do is called bootstrapping. So what that means is you pick 100 isolates by random. So you could either do it as false, meaning without replacement, or true, meaning with replacement. And we do this 1,000 times, and we can start to see a trend across time. So as you can see, I have looked at the chicken isolates in the beginning, but we don't have much data before 2010. So that's why I have focused on the human stool isolates. And we can see this increase across time. So the I is, again, the resistance across time. And so this kind of we can say that despite of the growth promoter ban in 2006, we can see that resistance is increasing. But is this the same case for the different strains? So this then comes into my second analysis, which is clustering. So what we can do is we have data for, again, on the x-axis is the ears and y-axis is the different clonal complex. The color indicates the probability of, so from 0 to 1, basically we're identifying if it is 1, it means it will be resistance. So for example, 2011 in clonal complex 464, it's always been, there's only resistant isolates. So we can see there are 3 main clusters: the resistant cluster, susceptible cluster, and mixed cluster. But this is a cluster used across time. So what about trend? So that comes into logistic regression. So as you can see, when we are actually looking at the time of resistance across time for different clonal complex, there's much more diversity. So I have made it a bit easier to illustrate. So also the red line is 2006, when the growth promoting ban came into place. So we can actually see about 6 different patterns of resistance trend. So we have 464 that actually we can't find any isolates before 2006, and it's only resistant. Or very, very highly resistant, where we have 353, 354, which has this kind of increase across time, kind of really nice sigmoidal shape, which is kind of scary as well. And we have other type of, for example, kind of this slow increase over time, such as the generalist 21, but we also have some that hasn't had much change, but in the middle, and some seems to even decrease a little bit. And we still have that susceptible cluster. So now we can see that despite in the beginning, we see that Campylobacter jejuni has had an increase across time, we actually have 6 different type of patterns just within the this bacteria. So one thing that I was really kind of interested to examine a bit more is this clonal complex 353. So we have this big change across susceptible to resistance. So I dived into this a bit more, and I've done different type of phylogenetic analysis. So one of the main ways is to use neighbor joining trees. So this kind of gives you a chance to understand the kind of the relationship between different different clonal complexes. So when Maiden Lab has made the clonal complex and kind of the MLSD scheme, they have already identified that there is 21, 48, and 206. So these dark blue in the middle, the light blue, and the green, which are also found in a couple of other places, to be kind of a clonal complex that is in a question, if you can say that's the word. But when it comes to clonal complex 353, this is something that we haven't mentioned before. So what we can see is that they're actually found all across the tree. And so this came to a question: is this the same strain? Are we actually finding some that are always susceptible, some that are always resistant, but we have kind of classified them into clonal complex 353. So is the central genotype correct? So this is something that I've been working on now. So one of the main things is between all these 4 groups, the 5th one is just 1 isolate. We can actually see that these isolates are coming from all across time, so from the 1997 to 2018, and they're all separate STs, and all coming from these main 3 regions: Oxford, Newcastle, and Aberdeen. But the group 4 has had a very different category, so it's all from ST581. And another thing that I really wanted to understand was, do we see the same trend across time? So in terms of 3 and 5, because they're very small isolates, we can't do logistic regression, but we can still see that 1 and 2 has a similar trend across time. So this kind of indicates that despite of them being in different places in the tree, we're still seeing that trend. So it's not completely different isolates, completely different strains, sorry. But group 4 is really, we can see that has a very different type of characteristic. It's completely susceptible across time. So I have done some clonal frame ML, and we can actually identify that 581 is completely different kind of strain. So we have actually now reclassified this into a new clonal complex called clonal complex 581, where we're writing a paper on this now. But when it comes to 353, on the other STs, we can see that the group 1 and group 2 has similar changes in resistance. So it's not necessarily that these are different strains or we have misclassified them. So just to summarize, so I guess in the beginning, we started off by saying, okay, we had this ban of growth promoter, but how is the resistance changing across time? We can see that there was this increase across clonal Campylobacter jejuni across time. However, when we actually dive into one layer down, we see that we have 3 distinctive groups for when we cluster them across time when it comes to resistance. And then we looked into logistic regression to see the trend across time, and that actually shows us there's 6 different type of patterns. And then to finish off the clonal complex 353, it's not many isolates, but 18 isolates have been reclassified to clonal complex 581. And we can see that trend is similar between 1 and 2 groups, but we need to further investigate why they're in different places. What can we do about them? Yeah, so just wanted to thank everybody, my supervisors, Martin, Mike, also Fran, who will be speaking to us today in the afternoon, and to the FSA, Anthony Wilson, and also to the organizers, and also to Ben and Evangelos, who has been helping me with some of the analysis, and the Chicken Journal Club as well. Thank you.
Okay, thank you, Desi, very much for this interesting insights about these resistances. We're already a bit advanced in time, but so there are lots of questions in Discord, so later in the Q&A we can answer them, but I will read maybe one from Tom Wilkinson. What do we know about fluoroquinolone usage during the same period, the data you present, and in the local areas you mentioned?
Yeah, it is a good question. Yes. So in terms of kind of for the each farms, we actually don't have access to that data. So if it is for therapeutic reasons, such as, for example, if there's one flock that has some kind of a disease, they wouldn't just they wouldn't just take care of that one flock. They will actually be given to, the antibiotics will be given to the water. So these kind of information is actually really difficult to get. Another thing that maybe I could try to do is to look at the sales of the antibiotics in the UK across time and kind of cross compare them, which I haven't done yet. So, but that is a really great question.
Okay, thank you very much again. And then more questions for you later in the Q&A.